also every vertex. And you have those constraints here, which are called subtour constraints. I'm going to talk about them in a minute. So this is a formulation presented by Meno, and the difficulty about this formulation is that, well, you have a quadratic objective. These are very difficult. If you give this formulation to a solver, it's going to have problems to solve this problem. So you have the quadratic objective multiplying binary variables, and you have this kind of constraint here, which is also kind of difficult. But then I can define a subproblem. If you let, actually, I'm saying here, if you already know the sequence of visiting, then you don't need those binary variables here. If you already know the sequence, you don't need to decide the sequence. You don't need those variables here. So if you have a fixed sequence of vertices to visit, then you just need to find what I call hitting points, which are the exact locations where the tour touches, visits the vertices. So you just have to find the coordinates in such a way that the total length of the tour is minimized. So you can write a second order common programming formulation for this subproblem. This has been shown by Meno as well. And then you can see here in this formulation, I don't have any binary variables anymore because I already know the sequence. So I have eliminated the combinatorial decisions. Now I just want to minimize. So zk here represents the distance between the distance of the tour visiting the vertices, and then you want to minimize that, the total distance of that tour, and then you just have to be inside the covering region, inside the disk of each vertex. Of course, you can generalize. I mean, you can write this formulation for the 3D case as well. You just need to add one more component here for x, y, and then you have to add another component here for z as well. So it's very easy to generalize to the 3D case. All right, then you can jump straight to the Ventricular formulation. Sorry, for the Ventricular algorithm. And we start this by defining a relaxation of our distance matrix. So in the TSP, you have the location of the vertices. For example, I, J, and K. And then you can compute the distance between those vertices. And you can call that dij, for example, dik, and djk. And then if you have many vertices, you can compute a matrix with all the distances here. You cannot do that for the cluster knot because you don't know where you're visiting the vertices. So I might visit this one here and this one here, and then the distance is going to be this one. But I can also do this one here and this one here, and the distance is going to be this one. So you cannot define a cost matrix for the cluster TSP. So we are defining we are calling this a relaxed cost matrix, which gives you a lower bound on the distance between any vertex, any pair of vertices actually. So you just need to compute the closest distance between any pair of vertices, and then you call this one lower bound for the actual cost between the actual travel distance between those vertices. So I do that, and then I call this matrix C min. Then again, I'm going to use the subproblem here. Let the sequence S, which includes a lot of vertices here, be any feasible tour. Sorry. So let S be any feasible tour over the vertices. I can define a function. I'm calling it SOCP of S, which computes the real cost of this sequence S. And then I can define a variable delta t, which is equal to z of t minus the relaxed cost for that sequence. So this variable is going to represent the difference between the real cost and the relaxed cost. Okay, so having that, then I can write this mixed integer nonlinear formulation for the problem. And then in this formulation, we have a linear objective using that relaxed cost plus delta. And then delta, this variable delta is defined here in this constraint, where you have that delta is greater or equal than delta t minus some big constant m of t, m at t, times n minus the sum of all arcs or all edges in that sequence. So what it does is I'm hiding this computation of z of t here, which is this guy here. I'm hiding it in a variable z. So this variable is here in the definition of delta t. So therefore, we are hiding z of t here inside of delta t. And this helps me to remove the nonlinearity of computing this guy here. So now I have two kinds of, I have two separate problems. I have this problem here, which, sorry, this is not nonlinear, this is actually linear. I have a mixed integer linear formulation here for deciding the sequences. And inside the computation of delta t, I'm hiding the nonlinearity here. Okay, but it doesn't help if I have to define delta t inside my formulation. So if I have to get this definition here and add here as a constraint in my problem, so to make sure we are respecting the Kuznets constraints, then I have no advantage in this. So what I do is, okay, now I'm going to remove some constraints of my original problem. And then I remove these constraints here. Because those guys here, they are actually nonlinear variables. And then every time I find an integer solution for this problem without those constraints here, then I can add one constraint for this particular sequence. Okay, so this guy here, I think we are done with this guy here. Now let's talk about those guys here. Those are the subtour constraints. And what they do is the following. If I relax this constraint here, the integrality constraint of my binary variables, then I'm only left with this objective here and those constraints, and this one. And this is a assignment problem, very well known problem also in combinatorial optimization. So what happens is, if I do that, if I remove those constraints here, these constraints here, I might have a solution like this. I have several subtours. I'm respecting the visiting constraints here, the assignment constraints, because for every vertex here, I have exactly one edge coming into the vertex and one edge leaving the vertex. But obviously, this is not a valid tour because I need a single tour visiting all the vertices at once. Okay, so I actually need those constraints here in my problem, but unfortunately, I cannot add them all at once, because if you check the definition of those constraints, I have for every subset S, so for every subsequence S, inside my set of vertices, then I have to consider every single S dash here. And the number of sequences here is exponential. So if I consider all of them at once, this is just going to be too many. There are too many constraints here. I have an exponential number of constraints here. So what we do is, to fix this problem, we can add these constraints one by one as long as we need them. So what those I just rewrote them here, and what those constraints basically says is that, okay, this guy here cannot exist. You cannot have 3 edges here for this set of 3 vertices. So you forbid this guy, and then you can forbid this one, and forbid this one, and run the algorithm again. I mean, run the relaxed problem here. But now we have some constraints here. Only the ones I need, not all of them. And actually finding these constraints, finding the right constraints here, which is, we call this the separation problem. This is called, this problem is reduced to a mean cut problem, which is, and its dual max flow, which are very, very well known in optimization as well. So I have broken down my very complicated problem in a series of simpler ones. Okay, so the overall idea here is I relax the delta and subtour constraints, so the red ones, and then I solve the relaxed mixed integer linear problem. Sorry for the N here. I use the min cut and max flow procedures to generate cutting planes, so to generate those constraints one by one as long as I need them. And then once an integer solution is found, I can compute delta and add a delta constraint in the relaxed problem. Then I repeat steps 2 and 3 until I have explored implicitly all the possible solutions. Not all of them, but only the important ones. So at the end, the objective function value of that of this formulation here. So at the end, this guy here should be equal to the optimal solution value of the close enough TSV. So here I'm presenting some results. Here I have some instances with 6, 8, 10, up to 20 vertices. And as you can see, if I go up to 16 vertices, I'm okay. Under 30 seconds, I can solve this, find all optimal solutions. But then if I go a little bit further to 18 or 20, then I take much longer to find all optimal solutions here. Okay, so now I'm going to present the branch and bound algorithm, just a different algorithm for the same problem. And the idea here is that there is an insight actually in this algorithm, which is in a TSP problem, since I have to visit all the vertices and I have to visit all the exact location of the vertices, then the tool needs to turn in an amount of times that is equivalent to the number of vertices. So I have to come here, and then here I have a turn here, and then I have a turn here, I have a turn here, and I have a turn here. But close enough, since the vertices they can actually overlap, I might not need to do so many turns actually. If I define one visiting point here, and I visit this point, for this example, I'm visiting all the vertices. So we are going to explore this idea here in the branch and bound algorithm. So now we split again the problem into two different problems, which is finding the sequence and then finding the hitting points or the coordinates where I'm going to visit the vertices. So the main problem here is going to be finding the sequence. The sub-problem is going to be computing the cost of the subsequence. Okay, so just a little example how the branch and bound works. I start here. You can see I have the dipole here. This is the starting vertex, and then I have all the vertices I have to visit here. I can start with any sequence of three vertices. So I have here 0, 3, 6, and 0 again. So I define this sequence here, and then I can compute for this sequence the optimal tool, the optimal distance. As you can see, by visiting only those, by adding only those points to the sequence, I'm visiting point vertex 4 already, because the tool is crossing this vertex here. So I don't need to add this guy anymore to the sequence. And then I have candidate, I have to define candidate vertices to be inserted into the sequence. And these guys are going to be the ones that are not visited by the current tool, which are 1, 2, and 5 here. So I have 1, 2, and 5, and then I have to add each one of them in each position, each possible position here in this sequence. So if I do that, I'm creating 3 child nodes. So I'm starting to build a tree here. A tree, sorry. I'm starting to build a tree here. Checking all possible insertions of the non-visit vertices to the current sequence. So for example, for this sequence here, 0, 1, 3, 6, 0, which is 0, this one here, I have 5, which is not visited. So I only have to add 5 here now to this guy here. Okay, so I do that for all possible combinations, but actually I can discard some of them based on branching rules and branching strategies. So if you are from the optimization community, you know branching bound already, you know how it works, and you know you don't have to consider all the branches. You only have to consider the important ones. So that's basically how the algorithm works. That's a very simple algorithm, actually. We developed 2 different branching rules, one based when the vertices have the same radius, and one for when the vertices have different radii. For the first case, I'm going to skip this a little bit. You just compute the distance from the center to the current tool, and then we add the one which is furthest away, which would be number 1 here. For the second one, if we have a number of candidate vertices to enter the sequence, the current sequence, we compute the contribution, how much that vertex would add to the objective function if I add that guy to the sequence. So for example here, if I add vertex 1 to the sequence, I'm contributing with this much to the objective function. If I add this guy here, I'm contributing this with this much here. And then I just have to compare gamma 1 and gamma 2, see which one is larger, and then I add the corresponding guy to the sequence. So there's a proof of correctness of this algorithm. This is published in Inform's Journal on Computing. The correctness means that the algorithm provides valid solutions and optimal solutions for the problem. And then now we have more computational results for this. You can see now that CPU times, actually I have another table here, comparing the branch and cut approach with the branch and bound approach. And you can see that the branch and bound approach is much more efficient, even for the largest instances. In fact, both approaches here, they are more efficient than the current ones in the literature. And moreover, the branch and bound approach, up to this day, it's still the state-of-the-art algorithm for the clustering of TSP. Even though it's kind of relatively old, so to say, it's from 2016. Then we can go for larger instances. Now we don't need to worry about 6, 8, 20 vertices. Now we can solve for hundreds of and thousands of vertices. This depending on the radius of each vertex. So here I'm comparing my solutions with the algorithm by Menel, which is a heuristic algorithm. Just keeping the numbers here, don't need to worry too much about that. Then I conclude my talk. I think I'm on time already. I have presented 2 exact algorithms for the clustering of TSP, branch and cut and a branch and bound. They performed quite well for these problems. As I said, the branch and bound is still the state-of-the-art algorithm for the clustering of TSP. Promising avenues of research includes new approaches for defining the root relaxation for the branch and bound algorithm. Up to this point, we start with only 3 vertices. And then you compute that tour. That's our starting root node in the branch and bound tree. But maybe you can develop better ones, which provide a better lower bound and speed up even more the computation. And here just some pictures. I just wanted to show some pictures because they are nice for a few instances with 20 vertices, and then have instances with 300 vertices here. And obviously we are exploring actually we are exploiting this the size of the radius of each vertex here to build a tool in an efficient way. Then you have with 195 And then you can also solve very easily generalize the algorithm for the 3D case here. We do not have circles anymore. We have now spheres. Then you have 100 spheres, 400, almost 500 spheres, and 1,000 spheres. And that's it. Thank you very much for your attention. Thank you again for the opportunity to present my work here. And if you have any questions, please feel free to ask them.
Thank you very much, Assistant Professor Walton Pereira Coutinho, for giving such informative and interesting presentation. Ladies and gentlemen, now we come to the question and answer session. Is there any question for Associate Professor Walton for his presentation? You could write your question on the chat room of our Zoom, or perhaps you want to speak or ask directly to him. You can write your name so our host can let you speak directly to Associate Professor Walton. Bagi para peserta, okay. There are two participants who raised the hand. Pak Hafiz, you may ask.
Bu Nahlia first.
May I?
Oh yeah.
Okay. Directly.
Hello.
Yes.
Yes,
okay.
Please.
Yes. Thanks for the change. Mr. Welton, it was a nice presentation, but absolutely I'm interested in the graph too about the shortest path. I want to ask you about the data that we use to manage. How you, what you call it, what kind of approach that you use to make sure that the data was show the real in the real situation. Somehow I usually use an approach, and however, how could the algorithm when the data not fixed or not what you call it, as real as the condition in the real life, our algorithm or our result was just passed by, can be directly used.
Okay.
Sorry for my English.
No, that's okay. I could perfectly understand you. So you're asking about this delta here.
Yeah.
How...
No, not the delta, but the data.
Oh, the data, the
yeah,
distance.
the data.
Oh, okay,
Yeah.
okay. Sorry. Sorry. So in this case, you are just in. Let me see if I can show this. Okay, this one.
Mm-hmm.
So in this case, for the particular case of the closing up TSP, this problem is already defined in the Euclidean space. So all the distances are Euclidean distances, and then you can compute them very easily. But for example, if you want to let me go back to this picture here. This is the one. If you want to consider, for example, the real distances between vertices here, and then you want to say that the distance between, let's say those are cities, and you want to know the real distances between these cities. I mean, you can just use like normal tools, normal map tools, and then build the cost matrix here, which would be the distance matrix here, D,
Then it
using the
means
components as the real distance between those guys here. And
then it means we use the real data.
Yes, you can use that, but that's not the case for the Klasnerf TSP. In the Klasnerf TSP, we just use Euclidean distances. I don't know if I answered your question. Okay, thank you very much.
Thank you very much.
Thank you.
Thank you, Walter and Nalia, for the question.
Latifah.
Oh, okay. There is a question from Pusapti.
Yes.
Can ask directly, Pusapti, please.
Okay. In determining the optimization of real problem modelings, TSP variant are often encouraged, and the discussion of no exact algorithm be developed to TRP variant, TSP variant.
Oh, sorry, I couldn't understand. I think my sound is a little bit.
Yes.
My sound is not great.
Bisa diulang pertanyaannya?
In determining the optimization of real problem modelings, TSP variant are often encouraged. Can the discussion of exact algorithm be developed to TSP variant? The discussion.
I don't know if I understood your question. Sorry, my sound is not great right now, but I think are asking which are the real applications of this exact algorithm for this problem, is it?
Bu Sapti, pertanyaannya itu tadi, I'm sorry, Waltong, I will ask her in Indonesian first.
Oke. Pada penentuan optimalisasi pemodelan masalah nyata, yang sering dijumpai adalah TSP varian.
Ya.
Nah, pada pembahasan eksak algoritma ini, apakah bisa dikembangkan untuk TSP varian?
I see. So the question is in our discussion here is typically or usually about TSP variant. Associate Professor Watson. So her Pusapti's question is it your CETSP, is it can be developed from the TSP?
TSP varian.
TSP variant.
Can I help? Can I help, Bu Latifah?
Oh yeah.
Yes, okay. I would like to translate the question from Bu Sapti. So in daily problem, we usually use the variant of traveling salesman problem. Okay, Walton. And is it possible to develop your optimization method into the variant of traveling salesman problem, like traveling salesman problem with time windows or else?
Dynamic.
Okay. Thank you for it. That's a very interesting question, actually. Thank you very much. So basically, for this problem here, for this algorithm here, which is the Ventricular algorithm, if you forget about these constraints here and about this guy, everything about delta, if you forget everything about delta, this is the standard Ventricular algorithm for the TSP. So for any TSP, you can use this algorithm, and then you can, of course, think about time windows and add more constraints here. This separation procedure is going to be exactly the same. We are not going to change it. So in the CUSNAF, you add these delta variables and delta constraints here. But for example, for the TSP with time windows, you might completely remove those guys here and add the time windows constraints in this problem here, and then you can solve it just as it is using the same exact separation procedure here, and use exactly the same separation problem here. So depending on your variant, if you're considering, for example, time windows, pickup and delivery, and other variants, the separation is basically the same. This is not going to change a lot, at least. So for the branch cut, this is the case. This is not the case for the branch bound algorithm. This one is especially tailored for the CUSNAF TSP because it takes advantage of this fact here, which you cannot. You just need to use a limited number of turning points to visit a large number of vertices. So actually, if you check the solutions here, let me go further a little bit. You can see that the number of turning points, for example, we have almost 200 vertices here. The number of turning points is very little. We have very small number of turning points here. So this is a characteristic of the branching bound algorithm, which is being actually characteristic of the problem, which is being explored or exploited by the branching bound algorithm. For the branching cut, then you might use these exact methods with a little modifications for many TSP variants. I hope I answered your question actually.
Okay, Bu Sapti.
Okay, okay.
Okay, thank you very much.
Thank you very much. Any questions from the other participants who want to ask directly to Associate Professor Walter? You may click the raise hand button.
May I ask a simple question? Bu Latifah?
Please.
Yes, please, Pak Hafiz.
Yes, so I noticed that for the distance, you present at one slide that the distance between two circle is the EIG, or the distance. It is the
This one?
yes, this one. CIG is the minimum distance between these two circle, I and G. But in another slide, when you have 3 or 4 circle, next. This one. I see that the distance from circle 1 and circle 3, I think it is not the shortest distance. And would you like to explain that?
Oh yes, sure. So here, let's have a little bit, come back a little bit. So here, this is for the branch cut algorithm. I start with this relaxed cost matrix. So I define this CIG mean, which is the minimum distance between I and j. But then every time, if you analyze this constraint here in the objective function, you're going to see the following. So every time we find an integer solution, which is a feasible sequence, feasible tool, then you're computing the real distance using delta t, because delta t has z of t here, and z of t is the solution of the subproblem. And this subproblem computes the real distance between two vertices. So what I do here is every time I find a feasible integer solution, this guy here is eliminated, and then I have the real cost, which is delta. And this is for the branching cuts. I have to do this because I don't have a way to compute the real distance matrix here. For the branching about is a little bit different because I don't have to compute the distance matrix at all. I never have to compute this distance matrix. The only thing I have to do is to compute the real distance for a given tool. So, for example, this slide here between 1 and 3, in fact, this is not the minimum distance between vertices 1 and 3, because the minimum distances would be something like here and here. But this tool here is the minimum distance tool visiting all the vertices. So maybe if I move my tool a little bit to this direction here, to be like here, then I'm increasing the tool of the length of my tool. Actually, you can see this red part here is longer than this blue part here. So that's the trick. Actually, in the branching cut, we use that distance matrix as a way of approximating the minimum distance of the tool, but that's not the actual minimum distance within the tool. In the branch about, we don't need to do that. We can directly compute the minimum distance of a given tool. I hope I answered your question.
Yes, yes. Thank you. Thank you,
Thank you.
Walton. Nice presentation.
Thank you
Okay,
very much.
thank you for Pak Hafiz for the question. Unfortunately, our time is limited. For participants who want to ask more about the topic from Associate Professor Walton Pereira Coutinho, you can contact him on his email on the curriculum vitae. Ladies and gentlemen, we finally come to the end of third preliminary session of ICoMathApp 2020. We would like to say thank you again to Associate Professor Walton Pereira Coutinho for his informative and interesting talk, and to audience for your active participation. Hopefully, the presentation will be beneficial for everyone. Thank you, Associate Professor Walton. We are honored to have you today. Good morning here.
Thank you very much.
Good night at your time. Wassalamualaikum warahmatullahi wabarakatuh. And I will pass the time back to our MC, Ms. Rafati. Thank you.
Thank you. It was my pleasure.
Thank you.
Thank you.
Thank you so much, Bu Latifah. And once again, ladies and gentlemen, let's give a big hand for Associate Professor Walton Pereira Coutinho from Federal University of Pernambuco, Brazil. It's a very informative and interesting presentation. Well, before we proceed to the next plenary session, there is an announcement from the committee. You can read it there in the chat box. I'm going to read it for you, that the presenters of the parallel session later should share their slides during the parallel session by themselves. So you will have the control over the screen sharing. However, it is recommended for all of you, the parallel presenters, to upload the slides or the PowerPoint in the following link. So that link that you can find in the chat box is a Google Drive link so that you can upload your slides or PowerPoints there. So in case there is a technical problem, we can access it there and share it for you. And please, once again, fill in the attendance link there in bit.ly/attendee D2. And you can just click it there in the chat box. Okay, now let's proceed to the second keynote session for today. We would like to invite Ms. Aziza, SPD MSI, as the moderator, and Ms. Aziza will lead the session delivered by Ibu Marlina Dumasari Nasution, PhD from Parallax International, USA. Ibu Aziza, over to you.
Okay, thank you. May I start right now?
Sure.
Okay. Good morning, ladies and gentlemen. Welcome to the last plenary session of the Second International Conference on Mathematics and its Application, ICoMathApp 2021. First of all, let me introduce myself. My name is Aziza, and in this session, I will be serving as your moderator. You will be hearing presentation from Ms. Marlina Nasution, PhD. But before we get started, I will read the brief biography of our distinguished speaker today. Pak Asmianto, would you like to share? Okay. Ms. Marlina Nasution, PhD, is a director of biostatistics at Paraxel International, Durham, North Carolina, USA. She has received her bachelor degree in statistics at Bogor Agricultural University and taken her master degree at North Carolina State University in statistics. Then got her PhD at North Carolina State University, also in statistics. Ms. Marlina's primary academic and research interests are biostatistics. She also joined several professional associations, and also she is the author and co-author of many articles in the fields of biostatistics. Ladies and gentlemen, before the presentation begins, I will tell you the regulation on how the presentation will be conducted. First, I would like to give the time to Ms. Marlina Nasution, PhD, to present the material for about 45 minutes, and then there will be 15 minutes for the Q&A session. And without further ado, now allow me to welcome Ms. Marlina Nasution, PhD, who will be speaking to us about statistics topic. Ms. Marlina, the floor is yours.
Thank you so much, Ms. Aziza. I will pull the slide text right now, shortly. Can you see my screen?
Not yet, Miss.
You can't see my screen?
No.
Only the explore internet what the folder.
Oh, wow.
Only
Yeah.
a
Let
folder.
me try again. Stop share. PowerPoints. Okay, this one.
Yes.
Okay. Yes.
And you know, great. All right.
Yes.
Thank you. Well, thank you, everyone. Good morning, everyone in Indonesia, of course. And this is evening time for me. It's 10 p.m. Excuse me if there is any sleepy face or things like that. But I'm so excited to be here around you. And according to the organizers, that I should speak Bahasa as much as I can. So I'll do my best to mix Bahasa Indonesia and English in this case to accommodate the organizers' request. However, my slide deck will be in English, in Bahasa English, of course. So before I start, of course, for everyone who's practicing Islam, Assalamualaikum warahmatullahi wabarakatuh. It's my pleasure here again to be here. Let's see.
Sorry, Miss Marlina, I think you need to unmute the microphone.
Yeah, thank you. I'm trying to make slideshow now. Slideshow from beginning. All right, so I'm back now. All right, so before I start, of course, again, I would like to thank Bapak Hendro Permadi yang sudah memperkenalkan saya dengan konferensi ini dan menginvite saya untuk berkontribusi. Terima kasih banyak, dan juga Bapak Muhammad Hafiz yang sudah berdiskusi atau berkomunikasi dengan saya mengenai update-update dari konferensi ini. Pembicaraan saya akan berpusat kepada biostatistics trends in clinical trials and how this COVID-19 pandemic impacts the trends itself. I'll divide this talk into the 4 topics. First, I would like to refresh everyone about what is a clinical trials and how these clinical trials challenges and being responded by all the people in the clinical trial arena before COVID-19 pandemic, and what is the direction after or during COVID-19 pandemic. And in the latest, in the final stage, of course, I would like to present the trends that I think it will be interesting for all of us here. Again, I'm on the side of the industry here, not academics, so it's more toward like practitioners' point of view. But you can see, and then you can try to implement that into the academical setting, of course. And these are all my own views. It's not necessarily present my employer views. So let's start with clinical research here. Clinical research or research klinis adalah part or a branch of ilmu medis or ilmu kesehatan tentunya, yang pada dasarnya mengkoleksi secara sistematis evidence. Evidence is facts or findings for new drugs to be established as a treatment. But actually, in reality, it's not just new drugs. It may also engage or involve new techniques or new medical devices or new methods or new therapy. So basically, it can be like more than just new drugs, but it's things that pertaining to the health of human or patients here in particular. Having said that, for this presentation, I'm going to use these terms interchangeably. I'm going to stick with new drugs to simplify. So basically, this research, it's to determine the safety and effectiveness of drugs intended for human use. And the ultimate goals for all of this, of course, to improve quality of life for human, in particular patients here. Again, patients first, patient first, patient first has been the emphasis of clinical research. Now, clinical trial itself is the experiment atau the observasi yang dilakukan untuk menunjang clinical research ini, riset klinis ini. It can be bersifat prospektif. It can be like in terms of like biomedical or behavioral research studies, or on human participants, or even like on human volunteers. It can be like patients, or it can be like a healthy human. If, for example, trials that engage vaccine, it will involve healthy human, of course. This will be designed to answer specific questions scientifically, of course. And based on that, it will generate data. It will give us data that can be used to assess or to evaluate safety and efficacy of the drugs on the humans, on the patients here. It can be in terms of like interventional trial, where basically the trial, the study is being set into like controlled environment, but it can also be non-interventional. So it's like mimicking the real life settings. So there is no specific drugs being randomized in this case. It is like non-interventional. And it is conducted only after they have received health authority. Of course, before everything can be started, it has to go through the approval from ethics committee. Of course, again, this is to establish the assessment of safety and or efficacy. Now, in the US, these clinical trials are divided into phases, starting from phase 0 through phase 14. Phase 4, sorry. But again, here I would like to emphasize that even though I including phase 0 here, walaupun saya include phase 0 is just an exploratory step that is not required as a part of the approval process of a new drug. So what it is being required are phase 1 through phase 4. So starting with, of course, smaller size of study yang kecil. Phase 0 can be like less than 15 patients, up to phase 1, where phase 1 basically untuk menginvestigasi apakah new drug actually safe. And when it is there is a finding that yes, it is safe, then it will continue to phase 2. That basically trying to investigate whether it's like this safe drug actually will work on human. And up to phase 3, where in addition to safety, there will also be efficacy assessment. Whether it's like this new drug is better than the control drug or standard care of treatment. And up to phase 4. So phase 4 basically is a phase where it is done after getting approval from regulatory agency. In this regard, is like FDA for USA. And the aim, the tujuan dari phase 4 ini actually untuk mengevaluasi long term of safety of this new drug. So before COVID-19 pandemic, it doesn't mean that there is no challenges on the new drug development. So in general, this new drug development are costly. It's costly and time consuming. So it typically takes up to 10 years, can be up to 10 years from preclinical to the approval of drugs, and will cost like hundreds of dollars for the development of this new drug. And even the cost of this research and development in pertaining of this new drug menjadi double between 2003 and 2016, based on some findings from some kind of committee, the Economist Intelligence Unit Committee. If the trajectory, jadi kalau pace-nya tetap, itu projection-nya adalah dalam tahun 2043, so around 20 years from now, the cost will be up to like even like more than $20 billion. We are talking about dollars here. So that's one of the angle, and that also because of that, a part of that, there is like low success rate. So basically because of the costly and time consuming, as well as there is a poor plan study designed to demonstrate the treatment effect, then there will be like a risk or a chance that the dose or regimen being investigated becoming like ineffective, as well as probably like patient population is not right one, so it becomes like ineffective as well. And in return, there's like lack of safety and or efficacy data, of course. And because of this complexity, it becomes unattractive, tidak menarik untuk pasien-pasien untuk mendaftarkan diri dan berpartisipasi dalam studi ini, sehingga mereka drop out dari study sehingga missing data dalam hal ini. Sebagai seorang statistician, of course we are concerned about missing data and not able to get conclusion from the data available. So there is, you can see that, like, of course at the end compromising the validitas of the statistic itself. And the last one, there is like stagnation in the development of innovative products. This is multiple angles of challenges that the new drug development have been known here. So based on the some report back in 2004, kita bisa lihat di sini bahwa ada penurunan dari jumlah aplikasi yang masuk ke dalam masuk kepada FDA here untuk new molecule entity as well as the biologic license, but at the same time, the costs are rise. So it's becoming the challenge now. Because of that, there are actions that being initiated by regulatory agency, FDA, EMA, of course, in other countries as well, China FDA, as well as Japan, India. But you can see here, bisa dilihat di sini, efforts yang inisiasikan membutuhkan waktu yang sangat lama. Dari mulai tahun 2004 here sampai kepada tahun 2019, baru ada final guidance untuk adaptive design. Adaptive design is the design that is like offer flexibility and can be utilized for more complex study, and as well as Bayesian. So there is like draft guidance on Bayesian as well being developed here to provide guidance to all the researchers in this arena, and also the pharma industry as well. So, in parallel, karena adanya dimulai adanya guidance, kita bisa lihat di sini ada sedikit kenaikan dari jumlah novel drug approvals up to 2018. We can see that there is like slight trend here up, but again, this is not like a speed or uprise increase, but it's like gradually increase. Now, what happened after the COVID-19 pandemic here? So recognizing there is like pandemic in March 2020, FDA and EMA bergerak cepat. Jadi waktu itu kan pandemiknya mulai March 2020, mereka June 2020 sudah mengeluarkan guidance for ongoing clinical trials, not specific to COVID-19. Any ongoing clinical trials. Basically, they emphasize that we need the researchers needs to ensure that the trial still meets the objectives, as well as keep up with the integrity, continuity, and interpretability, which means that they have to assess whether there is any impact of COVID-19 to their, for example, primary and key secondary endpoints. And if so, apa yang harus dilakukan? Kalau mereka harus modifikasi itu, misalnya jangka waktunya antara baseline sama tadinya misalkan 3 months menjadi 6 months, maka mereka harus menginkorporasikan itu di dalam study protokolnya, trial protocolsnya. So they have to amend that, as well as not just protocol, they also need to incorporate that in their statistical analysis plan. And this modification must take place before the final database lock and unblinding of the study. To avoid what? To avoid bias, of course. And in addition to that, they also emphasize the need of engaging independent team, that yang basically not the study team, but a team of experts that can help to evaluate or assess safety, any safety issue or efficacy even in unblinding mode before the trial becomes officially unblinding. And the FDA and EA also provide suggestions on mitigations as well for analysis, like you can see here. So the importance of capturing information that yang relevan dengan COVID-19 diantaranya adalah, for example, protocol deviations, right? Participants can deviate protocols. Mereka bisa, misalkan karena sakit, right? Mereka tidak datang ke visit yang harusnya mereka datangi untuk diambil ukuran-ukurannya. Tapi mereka tidak bisa datang, sehingga dengan itu berarti mereka deviate dari protokol yang sudah dicanangkan di situ untuk studinya. Nah, karena itu FDA dan EMA menyarankan untuk mengcapture itu. One potential solution is that to add a variable that basically flagging whether that event, that occurrence happens before or after COVID-19. That is just an example, simple example. So basically we need to make sure that we still in corridor. Kita tetap dalam koridor ketika kita mengukur treatment effect. Tidak terbiaskan dengan adanya COVID-19 ini. So after the pandemic, you can see here, so there is like website clinicaltrials.gov, you can search there. Ada banyak sekali melonjak jumlah COVID-19 trials. As of 2 days ago, there are more than 6,400 COVID-19 trials. COVID-19 studies, either like as a treatment or vaccine trials, basically, like finding COVID-19 vaccine. And of those, even like pivotal trials, which means that phase 3 trials that also needs to evaluate the efficacy of drugs, or in this case, like it's very important and then may require large sample. Now, having said that, karena banyaknya sekali studies, trials, we can imagine apa yang menjadi challenge here. We still not knowing about this COVID-19 100%. There's always evolvement here, right? Even like, for example, giving shot, there is like pro and cons, as well as the schedule of the shots. So things like evolving here. And also patient population, there's competition, high competition between studies. What we can do in the same time, there's also a hope that we can get cure sooner, right? So we need to like enroll fast. And we know that COVID-19 can like severe very quickly. So meaning that like safety review is likely to be done earlier by the independent team. That means that we have to be ready with that. We have to have the data, we have to have the outputs and so on and so forth. Again, like we have already discussed, there's other operational challenges like delays here, right? I mean, because of site closing, that impacts monitoring. There's no people available to get the data, so the data entry is delayed. Even like, for example, drug shipment can also be delayed and so on and so forth. So this huge amount of, or huge number or high number of COVID-19 trials have added another layer of challenges or wrinkles here. Now, let's go now to the latest trends. I'm trying to stick with the time. So what I see here as interesting clinical trial study designs that are being evolved right now are listed here. It's not, of course, inclusive list, but I'm only going to focus on the first two, adaptive design and master protocol. But the last three do not mean tidak berarti itu tidak menarik, tapi karena tidak ada waktu yang cukup. Jadi kita bisa diskusikan lagi nanti, misalkan. But for patient-centric, for example, basically patient-centric change the view of or perceptive of patients as passive to active as a participant, meaning that for this patient-centric trial, we also gain insights from patients on how this trial will go to, or what is the direction of this trial. So for example, patients can be asked for about visit, right, study visit, whether that's something that effective or things like that, or even we can like making sure that the endpoints, for example, involving patient outcome report. So basically it's like in engaging patients, so patient will be aware more into their own trial here. But then again, this patient-centric is relatively new. There are only like 5% trials now are patient-centric. But this patient-centric also can help with retaining patients or retaining subjects in the study, which means that at the end there are not many dropout or there are not many missing data. And this patient-centric somehow is like linked to that precision medicine as well. Precision medicine is another arena, but somehow it's like closely related to patient-centric, because basically precision medicine or personalized medicine is trying to choose the right treatment for the right patient and at the right time. So why we can do that now? Because of the technology. So because of the advancement of technology, we are able to identify, for example, biomarker, basically that molecule in our blood, right? That can tell us whether a body does the normal process or abnormal process that may indicate a presence of a disease. So that's why now personalized medicine becomes like booming, being boomed, and as well this has been backed up by the past president of the United States. In that case is Barack Obama. There was an initiative of precision medicine. Basically, it was for oncology, but it can also be applicable to so many other therapeutic areas. So again, like virtual trials, that's also another area that is very interesting. Different kind of challenges. It involves like mobile platform use and as well as wearable device. So there's a question about data privacy, for example, and how the regulation can be done for this virtual data, and as well as big data, real world evidence. So that's something that I believe that there are so many talks now discussing about this big data, right, in Indonesia. So that's combining that big data with the artificial intelligence algorithm. It can be useful for clinical trials. So now back to adaptive and master protocol. So recap, we do know that there are like challenges in clinical trials. So basically, we need to think about how we deal with the challenges, how we overcome these challenges. And one of those is by considering adaptive design. So adaptive design basically is trying to increase the efficiency on how we do the trials. We can by using this adaptive design, we can obtaining and accumulating information during a trial in real time. And as a result, reducing the cost and streamlining the timeframes for clinical trials in drug development. And with respect to how this adaptive design being applicable or being applied in the COVID-19 trials, now there are like more than 190 COVID-19 trials use this adaptive approach. You can see here there is like a trend of increase of use of adaptive design in so many trials. Up to like 2018, the data that I have is like up to 2018, but you can see that there is like increasing trend. And this graph also illustrates that the adaptive design itself actually includes bidang yang sangat luas. There is like adaptive hypothesis, biomarker adaptive, and so on and so forth. So having said that, again, I summarize this, the term of adaptive can mean so many things. Basically, it can be like in terms of like the form of randomization, adaptive randomization. So there's not equal likely here, but it depends on certain things, group sequential, sample size estimation, and so on and so forth. So I just want to like give you a flavor of a broad term of adaptive here. And even there's like some authors, including Bath and Mehta, has tried to categorize the adaptive design. This is something that you can look at in the article. This is a very nice summary. So you can see that again, it is really broad. It's from the early phase up to like the late phase of the clinical trials process. Now, based on FDA guidance, adaptive design clinical trials, basically it is defined the design that allows for prospective planned modifications. So prospective and planned modification, perubahan-perubahan yang direncanakan dan bersifat prospektif. Perubahan itu bisa satu, bisa lebih dari satu dari desain studi Anda. Dan kenapa this is prospective and planned modifications? Again, because we are trying to avoid bias, right? We don't want to like those compromise the validity, data integrity here. And the adaptation itself can be for the design, can be for the statistical procedures, and so on and so forth. Now, an example of adaptive design can start with a phase 1 dose finding here. Traditionally, phase 1 is to determine the dose and schedule of an investigational agent. Dose finding, sometimes also referred as a dose escalation. Two terms here, or two terms here that I would like you to focus on here is our maximum tolerated dose, basically the dose tertinggi that can be administered to patients safely. So basically the highest dose, safe dose to be administered, and dose limiting toxicity. Basically the dose where there is like side effect that can be tolerant for the patient. So that we consider it as like the event, the unacceptable or unmanageable safety. And usually it is based on the common toxicity criteria here. That is for the oncology study, of course. I would like to emphasize that the common toxicity criteria actually is like for oncology study, or cancer study, but like other criteria can also be used for other therapeutic area. And this is just an illustration. I'm going to skip this. This is a conventional dose escalation phase 1 design, which is called 3+3 design. So basically, based on this design, there is a pre-specified cohort size, either 3 or 6. And there is like a decision here. You can see, for example, if there is like, the first time always like 3 subjects being dosed, right, at the same dose level. And if there's like 0 DLT among those 3, then the dose will then be escalated to the next level of the dose. And if it's like there's like 1 DLT, then there will be additional 3 subjects. So akan menambah 3 subjects, kemudian dilihat apakah ada tambahan DLT. Kalau tidak, maka akan sama lagi masuk ke dalam route yang ini. Jadi kita bisa lihat di conventional design ini ada ketidakberbedaan, ada kesamaan walaupun skenario berbeda, kesamaan dalam mengambil keputusan, which is sometimes is not good, right? I mean, like, so basically that is one reason why this conventional dose escalation design, even though it is simple and easy to interpret, but people consider this as a design that has low probability for selecting through MTD. Well, we really need through MTD, right, to move on to the next phase. So there are alternative designs to this 3+3 design, like listed here. But one design that now lately becomes more and more being used because of the flexibility, because of the easy to understand, and in fact, actually, this design also has a special case, which is the 3+3 design. This is a combination of adaptive and Bayesian. Basically, they use adaptive and Bayesian. We can see here for this Bayesian optimal interval design. So this is the name of the design, Bayesian. For the Bayesian, instead of like yes or no, we have pre-specified boundary that we will compare with the observed DLT rate, where the observed DLT rate at the current dose will be the number of patients experienced DLT divided by the number of patients treated at the current dose. So this is a very nice design, and that lambda E and lambda D, lambda escalation and lambda escalate here, actually being derived from a Bayesian algorithm, a closed form Bayesian algorithm that is free of dose level and the number of participants. So it's really a simple pre-specified boundary. So this is the benefits of Bayesian. You can see this. This is just to summarize it. But again, you know, like this design is quite nice in compared to other similar design. And this is an hypothetical example. So basically, if you can see here, this is it is a case where the expected or the true DLT is at 30%. So that is the one that we are trying to estimate here. So supposedly there's like one patient being entered and then get dose at the dose level 1. So there's like five pre-specified dose levels. So there's like 5 dose basically, and then the total of 30 patients here. If the design is like 3+3, then number one is that there will be a difficult situation where where, for example, in this case, we assume that the patient number 3 achieve a DLT level here, experience DLT, and patient number 8 is not available. So because of that, there are only like 1, 2, 3, 4, 5 available patients, while the 3+3 design basically has to base the decision whether it's like escalate or no, based on 3 or 6 cohort size. So in this case, there is no way, or there is a difficult time for us to do that 3+3 design. But if it's like Bayesian, then we can go up to the next level and go down again here. If it's like 3+3 design, of course, we can also go up there. But then we will stop here. While for the Bayesian, we can de-escalate. There is an option of de-escalate and go up again. And at the end, if the design is based on 3+3, the MTD will be dose level 3, while using Bayesian, actually, the MTD will be achieved at the dose level 4, which means that over here, actually 3+3 is somewhat underestimated the MTD, because the 3+3 only use information from previous dose to determine the MTD, while actually the Bayesian use the across the dose levels information to determine the MTD. So it's more like flexible compared to 3+3. So that is one of type of adaptive design that being used here for the dose finding. And there is another designed seamless, which basically combined phase here, phase 1, 2, or 2, 3, 1, 2, 3, and so on and so forth. And again, this seamless design because of combining. There are like layers of challenges here that or considerations here that needs to be taught thoroughly, that includes well planned ahead, so that you can combine it without any interruption, of course, and statistical method that needs to be considered, of course, need to be able to potentially also include multiple looks in another cases, like interim, doing interim analysis into the data, and also needs to consider how we can combine the data from different stages right here. Two examples of seamless design are for this Pfizer COVID-19 vaccine trials. This phase 1/2 as well as phase 1/2/3. This the later study here, phase 1/2/3, actually the landmark trial that gained the approval from for the emergency use of from US FDA, so that people can get that Pfizer vaccine basically. Right now by January 2021, this phase 1/2/3 seamless design C4591001 actually has enrolled 46,000 patients. So that's basically how we can sort of like streamline or speed up the process of approval of designs, of course, without tanpa meresikokan safety and efficacy of the drug, of course. This is just an example, adaptive phase 2/3. Di sini bisa dilihat bahwa the end of phase 2 serve as an interim analysis, where basically the dose on the phase 2 are being selected according to specific criteria, and again also in addition to selecting dose, at the end of the phase 2, which serve as an interim analysis, there is also a re-estimation of sample size. So having said that, the next phase, the phase 3, because of like a seamless design, the sample size being re-estimated and having a new sample size and to start here, where basically this sample size calculation is being done using the weighted inverse normal method. Now, probably I need like 5 more minutes. Ibu Aziza, is it okay?
Is it okay?
So the last part of the latest trends is about the master protocol. So basically, this is an overarching or a huge protocol that comprise of several studies, sub-studies. You can see that how this master protocol is aimed, ditujukan untuk menjawab banyak pertanyaan, memasukkan berbagai objektif dari multiple studies. And this master protocol mempunyai banyak tipe. Here ada yang namanya umbrella, basket, platform. So basically different kind of objectives here. So in layman term, I would say this master protocol as a giant protocol comprising of multiple studies that relevant to multiple, either one or multiple targeted therapies in the context of one or multiple disease or disease subtypes. So basically in therapy, disease, and protocol here being combined, right? I mean, like the definition, in the definition. So this is the basket trial here. You can see this, the advantage and challenges of the basket trial, and the umbrella trial here, right? In contrast of to basket trial, the umbrella trial has multiple treatments, but single indication. It's relevant to COVID-19 landscape right now. And platform trial is another type of I would say umbrella trial, but the difference is that the sub-trials can continually enter and exit. So it's like, you can see here, for example, in the first stage, there are like 2 intervention and standard of care. But then in the next, after the interim analysis, the first interim analysis, 1 intervention is being dropped because of perhaps ineffective, right? Being dropped. But then a new intervention being added. Kemudian ada lagi interim analysis. Bisa dilihat di sini lagi intervention for is being added. So it can go away and go in at any time. So perpetual manner here. So again, there are of course advantage and challenges. We can see that how seamless and efficiency can be gained by platform trial, right? And it can accelerate the drug development. But at the same time, tantangannya adalah complex, of course. So there will be like tons of planning needs to be done. Team collaboration needs to be very good. It's large size, of course, and as well the scope as well. And the other thing that is interesting because of the perpetual manner of the enter and exit of the sub-trials, the randomization can be non-concurrent at some level, right, at some point. So we need to like think about that thoroughly. So how these master protocols being applied in COVID-19? So FDA just recently released guidance on how these master protocols are potentially be useful for studying the COVID-19, basically. And the guidance focusing on the umbrella and platform trials, which is like basically one disease, right? And per clinicaltrials.gov, actually there have been like 3 umbrella and more than 200 platform trials for the COVID-19 related studies now being done. So as a wrap up here, you can see, like the innovation part of master protocols. It's very promising using common screening platform, right? I mean, like screening criteria basically is common across the sub studies. So it means that we gain efficiency. Nggak usah harus dari scratch lagi again and again. Like if we have like separate trials, centralized and shared governance. For example, we have already I already mentioned about data monitoring committee. Kalau ada banyak trials berbeda-beda, berarti banyak tim juga, lebih banyak orang. Tetapi kalau it is centralized and shared governance, of course, there will only be 1 team of DMC needed. But again, needs to be like someone who's really like thoroughly thinking about commonalities as well as how consistency and coherence across the sub-trials within that master protocols. A common trial network, of course, it will be great. It will gain efficiency in terms of like the system, right? Insights, study sites, common protocol and design element, and so on and so forth. But at the same time, these efficiency efforts will require a lot of planning and coordination, again, like we mentioned before, and the need of the improved statistical methodology. And again, like we can think about changes in the marketplace, like at some point, the test treatment and a treatment can be a test treatment, but at a later time, that treatment can be approved already by the agency, meaning that that treatment can be considered as a standard care. So because of this, will require this master protocol will likely include a long term of years of studies. Then there is a chance that we have that changes in the marketplace as well. So we need to like think thoroughly on how we deal with that, from active being control later on. Aligning planning, design, conduct analysis. So there is like a framework that is called Estimans. Estimans is a framework that FDA is recently promoted here just to ensure that actually we measure precisely the treatment effect. So there has to be like considerations on how we deal with the intercurrent events, the events that is basically not related to treatment effects, but impact the treatment effects in any way. So that's the thing that among the considerations for use for these master protocols. And with this, I will conclude these presentations. I provide some references here that you can take a look later on. And thank you, everyone.
Thank you, Ms. Marlina. Thank you very much, Ms. Marlina, for giving such an informative and interesting presentation. And now, ladies and gentlemen, we come to the Q&A session or question and answer session. You can type the question on the chat room, or you can type your name or just click raise hand in Zoom, and I will let you to ask to the speaker directly. Atau bisa langsung bertanya dalam bahasa Indonesia, begitu Ibu?
Boleh.
Boleh, silakan.
Silakan.
Dipersilahkan kepada partisipan jika ingin bertanya dalam bahasa Indonesia. Oke, kalau begitu saya ingin bertanya kepada Ibu Marlina. Oh, ada Pak Hafiz, mohon maaf Ibu, ada Pak Hafiz. Ya,
boleh.
Bu Aziza terlebih dahulu.
Baik Ibu, saya ingin mengkonfirmasi juga tadi terkait dengan a point. Is it a model or just like the name of adaptive design or what? This is my first question, and the second, which part of Bayesian approach that yang digunakan that used in this model in point, which part of Bayesian approach? That's my question. Thank you.
Ya, so itu pertanyaan yang sangat bagus ya. Jadi untuk dose finding, phase 1 dose finding, or not just phase 1, it can also be phase 2. The design itu ada berbagai design. Ada yang rule based design like 3+3. Tapi Boyne itu adalah model based design. So answering that, right? I mean, it is a model design. So there is like certain models that even like I don't know in the details. I have to go back to the articles. But basically, that boundary that they develop pre-specified, like pre-specified. Jadi di awal sudah di-develop itu adalah based on Bayesian. There is certain model that they use. They call it as optimal interval. There is like I'm trying to remember it. I think it's like isotonic regression. I believe you can like double check, but yeah, that is the model that they use for that develop that pre-specified boundary. So I can look at here. So it is a closed form expression, and the lambda is engaging the logarithm. So basically the function of so there's like 3 hypotheses, right? Hypothesis that one is like relevant to when the dose needs to remain. Basically, there's no change. And the second hypothesis, like when the dose needs to be escalate, and the third one's like de-escalate. So there are like 3 hypotheses being tested at the same time. And from there, they develop the boundary that is a combination of those, the value at the hypothesis, basically the boundary itself. So for example, for the boundary for the escalation, it will engage that the value of escalation portion log 1 minus, like the portion divided by 1 minus the overall, I mean, when the decision needs to remain. So it's like divided by the log of those, like a ratio of psi 1 minus psi, like the multiplication of psi 1 minus psi. So it's like logarithm, engage the ratio of logarithm functions.
Okay, so may I say that the lambda is one of the parameters of that model? Is it
Yeah.
right? Yeah.
Yeah. Mm-hmm.
There are two lambda.
Yeah, there are two lambdas that is
lambda.
derived from the three hypotheses. So there's like psi and lambda here being engaged. So lambda is a function of that psi.
Okay, yeah. Thank you, Miss Marlina. Pak Hafiz, yes, Pak Hafiz, please.
Yes. I'm sorry for asking in bahasa Indonesia. Ya, nice presentation Bu Marlina. Terima kasih sudah berkenan hadir. Pertanyaan saya ini Bu, tadi kalau tidak salah itu ada tentang biomedis Bu, nggih. Atau, nah sebenarnya matematika kan kalau diarahkan ke sana itu kan banyak sekali wilayah yang bisa dijangkau dari segi statistik atau dari segi pemodelan. Nah, sedangkan mungkin permasalahan utama bagi akademisi di jurusan matematika sini itu Bu tentang akses memperoleh data. Nah, mungkin Bu Marlina bisa memberi gambaran kalau di sana itu seperti apa nggih Bu untuk mengakses datanya itu. Apakah di sana juga perlu kerjasama dengan rumah sakit atau pihak-pihak terkait. Kemudian sedangkan kan data kesehatan itu kan ya kita harus kayak ada izin tertulisnya ke pasiennya bahwa ini memang untuk data penelitian. Nah, mungkin bisa diberi gambaran secara umum saja Bu di sana. Seperti apa?
Ya, terima kasih Pak Hafiz. Itu sangat menarik ya bagi saya karena saya kan dari industri, dari research organization that mendukung pharma company basically yang actually the one that who owns this drug, right? That's being investigated. Nah, pertanyaannya tadi mengenai data. Ada data yang dipakai oleh academic staff, right? Ada data yang dipakai oleh industri. So, industri itu adalah environment that is very regulated. We have to follow standard operating procedures, standard procedures that have been governed every process of it, including bagaimana kita mendapatkan data, mulai dari pemilihan sites-nya. Sites itu apa ya? Kayak center-centernya di mana pasiennya ada, ya, basically pasien datang ke center itu nanti kemudian diambil misalkan yang easy, tekanan darahnya, berat badannya dan sebagainya dilakukan di center itu. Nah, data itu nanti di-capture, right? Di-capture. Tetapi untuk itu, supaya site itu meng-capture data, kita harus meng-include site itu di trials ini. Nah, untuk meng-include site di trial ini perlu izin-izin tertentu, apalagi kalau di negara-negara yang berbeda. So, pada prinsipnya izin itu diperlukan, betul. Makanya tadi ada yang namanya harus remember waktu ketika saya berbicara mengenai clinical trials harus ada approval dari ethics committee, harus ada approval dari country-nya juga kan. Jadi itu memang perlu proses-proses perizinan. Itu kalau dalam industri, apalagi kalau menyangkut hal-hal yang berkaitan sama development new drug ini. Tetapi kalau dari kalau the academic staff need the data, itu sebetulnya kalau ada kerjasama dengan industrinya itu sendiri, ya, dengan misalkan just say like because saya menyebutkan Pfizer tadi sebagai contoh, dengan Pfizer itu ada kecenderungan kalau memang sudah ada kerjasama itu akan bisa mengakses data. Tentunya datanya needs to be blinded, right? Tidak mereveal identity of patients and so on and so forth. So jadi yang saya lihat diperlukan di sini adalah kerjasama tadi. Memang kerjasama itu sangat penting. Di Indonesia saya rasa kan ada badan BPOM, right? Badan POM, right? POM, right? Ya. Saya rasa POM juga bisa menyediakan data-data itu untuk para akademia untuk melakukan riset, develop model or like working on design and then see whether design works or no, and then how to improve the design, things like that, right? Yeah.
Yes, terima kasih Bu. Thank you.
Okay, I think it's enough, Pak Hafiz. Okay. Ladies and gentlemen, well, we finally come to the end of the last plenary session. And before we close this session, I would like to take the conclusion from what the speaker have presented. The COVID pandemic has had such a wide impact, including in the field of biostatistic research. There are many challenges encountered, such as missing data, new factors, and also big data. One of the things that emerges to answer these challenges is adaptive design. And for big data, the existence of the artificial intelligence turns out to be very helpful. Ms. Marlina, thank you so much for giving us a very interesting presentation. And we also want to say that please don't leave this Zoom after the event is closed, because Pak Hendro Permadi has a surprise for you. And thank you very much, Ms. Marlina. Okay, Ms. Rahmati, I will pass the time back to you. Thank you.
Terima kasih.
Thank you so much, Ibu Aziza. And ladies and gentlemen, once again, let's give a big round of applause for Ms. Marlina Nasution. Well, honorable guests, ladies and gentlemen, that was the last session of plenary session of this conference. These 2 days, we've learned a lot from the 5 keynote speakers explaining 5 different topics. Once again, we'd like to say thank you for Professor Dr. Toto Nusantara, for Associate Professor Walton Pereira Coutinho, for Assistant Professor Marco Murandotti, for Ibu Marlina Dumasari Nasution, and Mr. Martinus Frederick Ezerman. Thank you so much. We're really grateful for you've shared your knowledge and insights and expertise. For the participants, thank you for the unwavering attention during the 2 days plenary sessions. And thank you so much for the committee for your hard work. Thank you to Department of Mathematics, Faculty of Mathematics and Social Science, and to Universitas Negeri Malang for all the support. Let's have a big hand for all of us. And ladies and gentlemen, the plenary sessions is officially closed. Let's take a picture together. Please turn on your camera and set the virtual background. The operator will help us to take the picture, and there are going to be 3 takes. I'm going to count from 1 to 3 and then smile. Okay, the first take, 1, 2, 3, smile. The second take, 1, 2, 3, smile. The last one, 1, 2, 3. Okay, thank you so much. We've got all of your pictures. Well, people say gaining knowledge is the first step to wisdom, and sharing it is the first step to humanity. Let's keep gaining and sharing knowledge, be wiser and humbler. Well, another pantun from me. Kids love geometry. They said it's fun lesson. Let's have a break, everybody. See you in the parallel session. We're going to see you at 10:15. So please be prepared and go back here again in around 12 minutes, I think. So in 10:15, we're going to see you here again in the parallel session, sorry. Have a nice day, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
For Ibu Marlina, please stay here. Yeah, because there will be a surprise from Pak Hendro, I think.
Oke, terima kasih Ibu Marlina. Kalau saya pakai bahasa Indonesia, Lin masih ingat saya. Teman-teman hadir, ada beberapa yang hadir. Masih ingat nggak permintaan Bu Marlina waktu semester 3 sama saya?
Waduh,
lupa. Lupa. Kalau yang minta, kalau yang nostalgia nih, kalau yang minta lupa, tapi saya masih ingat Lina. Oke, jadi gini aja, Fes, ada sesuatu yang pengen saya serahkan ke Ibu Marlina, gitu. Si Sauf, nanti kalau hard copy-nya mungkin bisa saya kirim ke rumah di Bogor. Lina di Bogor apa di US ini?
Masih di tempat yang sama.
Masih di tempat yang sama. Oke, silakan Pak Asmianto.
Oke, siap Pak.
Assalamualaikum warahmatullahi wabarakatuh.
Lagu ini saya persembahkan untuk Ibu Marlina untuk mengenang Bapak Profesor Dr. Insinyur Andi Hakim Nasution, ayahanda Ibu Marlina yang paling dicintai sama Bapak Andi. Lagu ini hanya sekilas saja tentang Bapak Andi Hakim Nasution. Mudah-mudahan Ibu Marlina berkenan menerima lagu ini. Oke. Andai ku bisa tulis tentang dia, naungan rembulan akan menerangiku, dan angin pun berdesir membisikkan tentangnya. Ingatan menjadi lantang untuk mengingatnya. Hanya Engkaulah Tuhan yang mampu menjadikannya angan impian dan harapan serta dambanya. Karya dan kiprahmu mendunia, impian semua anak bangsa. Masih terasa walau kau telah kembali padanya. Nasution adalah margamu. Andi Hakim panggilan namamu. Sosok yang pernah ku tahu walau sesaat. Untukmu ku doakan tempat yang terbaik, tempat yang Engkau janjikan indah dan tak terbayangkan. Oh indahnya, oh indahnya, namamu selalu ku kenang, namamu selalu ku kenang. Terima kasih. Ini untuk Ibu Marlina, mohon aransemen disempurnakan. Saya hanya memenuhi janji ketika kita masih kuliah. Ibu Marlina pernah memesan sebuah lagu, tapi saya tidak sanggup. Terima kasih Bu Marlina.
Luar biasa Pak Hendro, luar biasa.
1, 2, 3, tes.
Iya.
Saya berusaha menahan tangis, soalnya banyak orang malu juga menangis, tapi sangat luar biasa perhatiannya dan juga bakatnya yang terus menyala. Jadi luar biasa. Terima kasih banyak Pak Hendro. Tidak menyangka sama sekali.
Pak Hendro.
Iya Pak.
Saya juga termasuk orang yang pernah kenal dekat dengan Pak Andi Hakim Nasution. Pernah menjadi ketua tim Olimpiade. Saya sempat beberapa tahun, 3 tahun kalau nggak salah bersama beliau sampai 2002 ya beliau wafat sama salah. Saya juga termasuk yang mengenang beliau Pak Hendro. Terima kasih Pak Hendro.
Terima kasih Pak Heri.
Terima kasih Bu Marlina.
Ya itu Lina, waktu itu Bu Lina minta sebuah lagu, saya nggak bisa. Tapi sekarang nggak tahu. Agak susah membuatnya, lebih susah kalau membuat artikel. Aransemennya saya nggak mampu. Semoga nanti dirubah Bu Marlina. Kalau misalkan disempurnakan, hanya itu yang bisa kami berikan. Saya juga mewakili jurusan matematik. Terima kasih sama Ibu Marlina. Mudah-mudahan di lain waktu bisa kita jalin kerjasama yang lain. Gitu ya Lin ya. Lina nggak usah nangis.
This is very invaluable, sangat-sangat berharga. Tidak ada bandingannya. Terima kasih banyak Pak.
Iya. Nanti kalau memang ini rencana saya cetak buat Bigora, lalu saya kirim di Bogor aja. Buat ini, mohon untuk alamatnya di Bogor, WA ke saya aja. Oke, Lina, terima
Terima kasih.
kasih. Ya, gitu-gitu. Salam buat keluarga di sana dan Ibu di Bogor, nggih. Ya,
Oke,
terima
terima
kasih,
kasih.
Lina.
Matur nuwun semuanya.
Ya.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi
warahmatullahi
wabarakatuh.
wabarakatuh.
Thank you so much, Pak Hendro. It's very wonderful. Sampai terharu yang
Baik.
mendengarnya.
Ya, dia mau menangis.
Thank you so much, Pak Hendro. What a very nice song. A scientist, a songwriter, and a singer. Very talented. Okay, ladies and gentlemen, as I've told you earlier, we're going to have a break, and the operator will play some songs while waiting for the parallel session to start. Let's enjoy the song and see you later in the parallel session. Assalamualaikum warahmatullahi wabarakatuh.
Pak Hafiz. Halo, selamat siang Pak Hafiz.
Oke, Pak Made.
Itu saya kirim file yang terakhir ya. Ada belakangnya revisian gitu Kak.
Oh, oke.
Ya. Terima kasih.
Oke.
Apa sih buat artikel apa?
Pak Mabul nggak lihat, Pak Mabul.
Good afternoon, everyone. The operator have set the breakout room, so please manually join the breakout room that you're assigned to. You can click the breakout room symbol in the toolbar and then just choose whichever room that you're assigned to, then click join. And then you will be automatically be in the small breakout room.
Hey yo, let's rap UF, let's rap more. Hey,
PKKMB Lenceng.
Ciro lupa. Selamat datang kawan di kota pendidikan, Universitas Negeri Malang. Belajar di sini untuk cita impian. Salam cakrawala, itu milik kita.
Jaya diri untuk raih prestasi, tunjukkan jati diri, wujudkan imajinasi. Bergerak selalu, tak pernah diam, giat berkarya untuk capai cita.
Wawasan luas adalah jawaban. That's right. Untuk menggapai masa depan. Pribadi yang bekerti dan saling menghormati pada setiap pribadi tak terkecuali.
Kecerdasan adalah kemampuan untuk adaptasi dalam perubahan.
Universitas Negeri Malang, berkarya dan terus berkarya. Universitas Negeri Malang, lambungkan asa menuju cita. Universitas Negeri Malang, inovasi tiada henti. Universitas Negeri Malang, lambungkan prestasi untuk negeri. Belajar itu penting, jangan dibawa pusing. Baca buku yang sering, walaupun ongoing. Semangat mencapai impian dengan belajar dan terus belajar. Walau rintangan dan silih berganti, tetap hadapi dengan hati yang sabar. Makan nasi rasanya hambar, hati boleh ambil, tetapi prestasi harus tetap ku kejar.
Pembuktian dan pengalaman, mampu dan adil untuk jalankan kewajiban. Tak kenal lelah, untuk pengalaman yang banyak, jujur dan bijak dalam bertindak.
Beradaptasi dengan sekitar, bagai mentari yang sedang berpijar. Jadi diri sendiri, gak perlu basa-basi. Kenali diri tuh, kembangkan imajinasi. Universitas Negeri Malang, berkarya dan terus berkarya. Universitas Negeri Malang, lambungkan asa menuju cita. Universitas Negeri Malang, inovasi tiada henti. Universitas Negeri Malang, lambungkan prestasi untuk negeri. Universitas Negeri Malang, berkarya dan terus berkarya. Universitas Negeri Malang, lambungkan asa menuju cita. Universitas Negeri Malang, inovasi tiada henti. Universitas Negeri Malang, lambungkan prestasi untuk negeri.
Aku room 5 ya.
Sudah masuk belum? Aduh, nek endi iki? Attendance, saya masukkan nek endi iki? Filenya. Belum. Presenter. Ya, berarti saya harus download dari ya, dari apa itu? Saya masuk ke.
Oke, Bu Santi.
Maaf, nggak jadi.